Hi ma'am, good morning. I'm Giselle Monquillo. I will report about genetic modification. Genetic modification is a technique to change the characteristic of a plant, animal, or microorganism by transferring a piece of DNA from one organism to a different organism. This is done through targeted removal of desired genes from the DNA of one organism and adding them to the other organism. So, genetic modification is a technology that involves inserting DNA into the genome of an organism. To produce a genetic modification plant, a new DNA is transferred into a plant cells. Usually, the cells are the ground initial culture where they develop into a plant. The seeds produced by these plants will inherit the new DNA. Three types of genetic modification method for crops. First, traditional crop modification. Two similar plants cross-pollinate or create a new plant. Traditional crop modification. Traditional method of modifying plants like selective breeding and crossbreeding have been around for early than 1,000 years. Most of the foods we eat neatly were originally created using a combination of traditional methods. So, traditional breeding is genetic trees have been selective and in some cases integration through crossbreeding of the same of related species in a traditional breeding. Genetically modified plant. The plant feature a novel genetic material combination that was introduced by modern biotechnology techniques. Second, genetic engineering. A gene in a soil bacterium is inserted into the DNA of the corn to create an insect-resistant corn. Genetic engineering is a method that, among other things, enables scientists to copy a gene with a desired trait in one organism and put it into another. Genetic engineering has been used since the 1970s and builds on the scientific advances we have made in the study of DNA. So, genetic engineering is a deals with the modification of genetic material, an organism and which genetic material of favoring to. We know that is the organism and what about of the remaining ones will and then is the RNA which has genetic material just like in the case of some viruses. So, genetic engineering deals with the modification of genetic material. Third, genome editing. One example of genome editing is removing an unwanted gene. Genome editing. Genome editing is a new method that gives scientists more precise and targeted ways to develop new crop varieties. Genome editing tools can make it easier and quicker to make changes that were previously done through traditional breeding. So, genome editing is a group of technologies that give scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at particular location in the genome. So, there are many applications of genome editing. The dream is to be able to cure people who have disease because one gene in his or her organism is not working properly. But there are also other possibilities. You could modify plants instead of introducing a lot of foreign material. Just change one gene so they would be resistant, for instance, to drought, or you could modify an animal to be less sensitive to cold or grow better This technique could really be revolutionary in treating diseases. It will not be cheap, it will not be easy, but it could change a lot of things. It may change the way we transplant organs. Special sort of pig has been made that would not infect us with a virus. If their organs were transplanted instead of very elaborate techniques to modify plants, you could have a very simple one. Genetically modified organism can be defined as organisms, plants, animals, or microorganisms in which the genetic material has been altered in a way that does not occur naturally by mating and or natural recombination. Foods produced from or using GM organisms are often referred to as GM foods. Organism whose genome has been engineered in the laboratory in order to favor the expression of desired physiological traits of the generation of desired biological products. In conventional livestock production of crop farming, even pet breeding, it has long been the practice to breed select individual of a species in order to produce offspring that have desirable traits. In genetic modification, however, recombinant genetic technologies are employed to produce organisms whose genomes have been precisely altered at the molecular level, usually by the inclusion of genes from unrelated species. Organisms that code for traits the world obtain easily through conventional selective breeding. What exactly is GMO? Genetically modified organisms are living organisms whose genetic material has been artificially manipulated in a laboratory through genetic engineering. This creates combination of plant, animal, bacteria, and virus genes that do not occur in nature or through traditional crossbreeding methods. There are 10 genetically modified foods: corn, soy, cotton, papaya, rice, rapeseed, potatoes, tomatoes, dairy product, peas.